I know there may be a few of you who may not be that familiar with my background, and so I thought I'd just kind of give you a little bit of information about where I came from and what got me here to the Center for Agroforestry. So I grew up in Illinois, and I got my degrees in Illinois, bachelor's, and I got a master's degree in weed science. So I was much more on the agronomy end than I was on the forestry end. And after I got my master's degree, I worked in industry for a few years, worked for what was called Zeneca at the time, and then became Syngenta, and then moved on. Went and got my PhD in agronomy as well, also focusing on weed science. For my master's, I was working on weed resistance to herbicides. For my PhD, I was looking more at degradation of herbicides in the soil. After PhD, I worked for Dow AgroSciences for about 3 years, and I lived in Iowa during that period, in Winterset, Iowa, and covered a pretty large territory that included Iowa and part of Illinois, and then got larger as I stayed there. And after that period, I kind of made a real kind of shift in my own thinking and was not feeling super great about where I was at with what I was doing in my career, so decided that I was going to go back to school, much to the chagrin of my husband over here, who thought I had enough degrees as it was. But so I went back and got a master's in landscape architecture. And then when I finished that up, my first faculty appointment was at University of Vermont, and I was there for 3 years, and then was back at the University of Illinois for about 10 years before I came here. So my new role at Mizzou here is, of course, as the director of the Center for Agroforestry. And so I have kind of administrative capacity with that work. I'm also co-superintendent of 2 of the research stations that we have, the Horticulture and Agroforestry Research Center and Land of the Osages Research Center that I'll talk about a little bit more. I have a teaching appointment as well. So this spring I'm teaching Ecological Principles of Agroforestry. These are both going to be online courses. So the spring one I'm teaching now is for graduate students. And then in the fall, my plan is to teach an undergrad level course on urban agriculture and agroforestry. And that will probably also be mostly online, but may have like one session that's on campus. And then I'll also continue my research in landscape multifunctionality. And just a kind of little bit of background on landscape multifunctionality, it's this framework for thinking about how we integrate ecological, cultural, and production functions, and really focusing on a better understanding of the landscape, the context that we're in, recognizing that there's something very different about designing something if you're in an agricultural context versus an urban context. And also really connecting to the needs of the users of that landscape. So they might be landowners or farmers or rural residents or urban residents. And so thinking about how we stack those different functions. And so if we're talking about functions, production functions are the primary component that we particularly think about with agriculture. So it can be producing food or feed or other types of materials, even like producing firewood or something like that could fit under production functions. Ecological functions tend to be more oriented toward the conservation end of the spectrum. So think about conserving biodiversity, nutrient cycling, carbon sequestration. And then the item that often gets overlooked is the cultural function aspect. And so there's this cultural dimension that we can also tie into this whole thing that can help people have a better connection with the landscape. And so it helps to kind of bring in the agricultural values, the land values, the conservation values. So thinking about recreational functions, visual quality of the landscape, artistic expression, even using the landscape for things like education and research can be valuable. So agroforestry is like the best opportunity to actually work on a multifunctional landscape. It just by very definition is integrating the production and the ecological functions, and certainly has a cultural aspect as well. So we're bringing trees and shrubs together with crops and animal farming and creating these multiple benefits. So it's really cool to think about how we use agroforestry in that multifunctional landscape context, and really thinking about a solutions-oriented approach. So there's a lot of focus on problems that we're facing, like Climate change and climate variability. Well, we see agroforestry as one of the best solutions to these problems. So in terms of the climate change piece, we're producing woody biomass that can store carbon. We have lower inputs so that less greenhouse gases emitted. Habitat that actually mimics some of the habitats that are lost. So part of the problem with this whole situation is that there are forested landscapes being lost. So if we're recreating those in a slightly different way, then we're offsetting those problems. We have more diverse crops that are going to be more resilient to climate, climate extremes. So if we are only focusing on one crop, we are in a very risky situation. And also from the social, kind of social justice perspective, we know that climate change impacts are going to be more problematic for certain communities, and probably those that are like the low-income communities, marginalized communities. So with these practices where we're trying to negate these problems, we can also have an important impact on social justice. Thinking about natural resource damage, also here, agroforestry takes a big role, protecting our water resources, really all of the things that you guys were talking about earlier today, cleaning the air, having these diverse systems that hold on to our genetic resources so that we can continue to draw on genetic resources for improving crops, retaining and building soil, using less fuel. And again, looking at kind of a social justice perspective, recognizing that some of these resources are on places like tribal grounds that are really sacred, important places. And so we need to begin to kind of build that into our thinking. And then finally, the food insecurity challenge that we face. Again, the agroforestry solutions look at we're producing foods in some of these systems that are directly consumable. So they could be more efficient. They don't, in some cases, don't require as much processing or transport. We can reduce those food miles. We have more diverse plantings, which means we're setting ourselves up for a more diverse diet. So that's going to result in a more healthy human. And also in terms of just the quality of the foods that we're producing. We're producing nuts from trees, berries from shrubs, and these are some of the most nutrient-dense foods that are available. Very important. And giving communities greater access to these healthy foods can help combat obesity and other diseases. Okay, looking at future plans and themes, this landscape scale perspective is something of great interest to me, thinking about identifying those marginal lands, trying to do suitability analysis, and thinking about planning at the whole farm level. Also, this idea of productive agroforestry, so really emphasizing the potential to bring in specialty crops into agroforestry. And even more specifically, like selecting crops for the human health, the human condition, things like dark berries that have just a real human health benefit. And then improving species for weather and climate. And then research plans looking at urban agroforestry as well. So that urban setting, opportunities to map opportunity lands in urban areas, trying to reduce the risk related to contamination, thinking about which species are actually well adapted to that urban condition. Just a couple of highlights here in the last year as we run out the clock here. We have a couple new faculty hires. I won't spend a lot of time on them because they're going to be presenting, but Dr. Ron Revord and Dr. Ashley Conway joined our group. We have a new agroforestry podcast series that Hannah will probably talk a little bit more about. And a big thing for us was the Land of the Osages Research Center, which was a new center, new research station that was given as a gift, and it also creates this connection with the Osage Nation. And so I'm just going to flip through a few pictures here. We had the grand opening on October 29th, which was an amazing event, brought in Osage dignitaries, and they did some of their ceremonial pieces. Chief Standing Bear and others delivered really amazing, heartfelt messages. It was a really special event. I think everybody that was there really felt that. So it was just a really neat event. And then finally, I'll end with this, the future launch of our new website. We're not quite ready to launch yet, but Hannah and I have been working on getting our website ready to go and working on the design and everything with our group within Ag Eb in our own college. So that's kind of exciting that it's been within that group. And a couple of the pages so you can kind of see how it's set up. That one's intended for landowners as an example. That's all I have, and we'll keep things moving.
I'm just going to talk briefly about the current state of our graduate education. We established graduate program and a formal emphasis area in agroforestry back in 2011, and it's in addition to the campus-based opportunity, there's also a fully online program. So just some numbers, update status. In the year of 2019, we had 11 students finish their graduate programs, MS, PhD, online MS, and graduate certificate. We currently have 48 graduate students enrolled, and I threw some numbers in there. A good number of PhD and campus MS, a lot of online MS, and a number of online graduate certificates, and 14 of those 48 joined the program in 2019, and we already have some new folks that are joining in 2020. Right now, the center is currently supporting 11 graduate research assistantships within the School of Natural Resources, but we do co-fund some programs jointly with other colleges and divisions outside of the School of Natural Resources. Get rid of that. There you go. And there's a large number of faculty that are involved in teaching these courses that have been. Many of them you're going to hear from. It's a long list in very diverse disciplines, but when you're delivering content in agroforestry, you really have to have a tremendous diversity of expertise to deliver a curriculum like that. In the year of 2019, we had 85 graduate students enrolled in 9 different courses, all online, as part of the agroforestry contribution. And we have new online courses that we're excited to see with some of the new faculty that have come on board, Ron Revord, Ashley Conway, and we're also going to have Chung Ho bring some of his teaching that's happened on campus available also to our online students. Short and sweet.
I'm the new tree breeder geneticist in the Center of Agroforestry. I'm filling the role that was formerly held by Mark Cogshell, who's done a lot of work on black walnut. And one of the foremost responsibilities of my job is to make use of the genetic resources that were left over by Mark. And so we'll talk about one of those projects today. So quantitative trait loci mapping of commercial traits in black walnut. So a little background on the black walnut research that the center has done. Started back in 1998, led by Mark in the upper right-hand corner there. And the leading goal being to domesticate eastern black walnut for orchard production. So a driving characteristic of interest there is increasing percent kernel size of the nut. The genetic base for this work was 64 cultivars that were more kernel production orientated as opposed to timber based. Mark's made 2 generations of offspring. You can get an idea of the size of those generations in the sub-bullets there. So around 1,000 full sib offspring have been evaluated at HARC over the years, and around 100 half sib families. You can, on the lower right-hand corner, get a sense for the pedigrees that have led to some of his breeding selections. Selections that the center is now trying to propagate and do replicated trials on across the state. Those would potentially be the first cultivar releases that have been genetically improved with intentional breeding that could go out to Missouri growers or those in neighboring states. So Mark also was very forward thinking. He didn't just have his applied breeding pedigrees, but he developed some genetic resources so that we can improve breeding practices going forward. One of those resources is 2 mapping populations. And so we see the 3 parents up there, Sparrow by Schussler and Sparrow by Emma Kay, and their relative sizes. He's worked with the University of Notre Dame to do rather deep sequencing of these individuals and their parents. You can see an initial linkage map presented on the right with about 1,300 DNA markers. They've done really deep sequencing of the parents, like whole genome sequencing, so there's great reference databases for annotating downstream. And they expect around 1,000 high-quality SNPs will be added to the linkage map over time, so there'll be very fine-scale mapping of traits. So now these trees are in about their 10th year, so they're coming into a stage where it's relevant to start phenotyping so that we could look at trait segregation and ascertain the suitability of respective traits to QTL mapping. A little bit of backstory that would contextualize that is these parents weren't necessarily selected and hand pollinations made to derive offspring based on respective contrasting traits. A lot of times full SIB families are reconstructed through genetic fingerprinting. So you'd collect half SIB seedlings or seeds from a given female parent, and then you genetically fingertype those seedlings and then reconstruct full SIB parents by figuring out who the pollen parent was. And so these two mapping populations were derived in that fashion, and so now we have this resource and we have some idea of parental traits, and we need to see which ones actually would result in good QTL mapping options. So that's where we stand here. So objectives in QTL mapping, we're trying to identify genetic regions where these traits lie, and then DNA markers associated with those traits of interest. Some classes of traits, so phenological traits, production-oriented traits, and disease resistance, particularly anthracnose resistance, and you could see the phenotypic response in the yellow leafing there, and then the necrotic areas. Some short-term targets, bud break and mean pistillate bloom date, very early season traits that are tangible and not complicated. We see lateral bearing. It's represented in the lower right-hand corner, where you don't just see fruit at the terminal end of a branch, but you see it along the axillary shoots of that lateral branch. So many more fruit per lateral branch. Some midterm targets relate to nut and kernel quality traits. So these plants are just now coming into bearing years. And then working with Chung Ho to elucidate some of the metabolic components of anthracnose resistance. So we've started to try to get perspective on the parental phenotypes and if there's going to be enough variation in these progenies for respective traits. And so a good example for phenology is bud break, and ample segregation is expected by looking at the phenotypes of the parents in relation to our germplasm base. And so each horizontal bar is represented by a cultivar in our germplasm collection. And you can see Julian days on the bottom where we have bud break occurring. And so Schessler is at one end of the spectrum, Sparrow is towards the other. They're quite distantly separated. It's almost 2 weeks in bud break time. Let's see, a little perspective on, so early bud break allows you to maybe avoid some stress during nut filling. Could be advantageous from that perspective. Late bud break could be frost avoidance. And there's some precedence for mapping bud break from the Persian walnut, which is the walnut that's been most prevalently studied. Last year, the UC Davis program discovered a single QTL for this trait. Looking forward, something like percent kernel, again, a similar type of chart to show you where these two individuals fall in, as far as the genetic base of this trait goes. So they are distinctly separated. Sparrow a bit more than Schessler from McKay, but the Sparrow McKay mapping population should give us a good chance to find a QTL for this trait. And you get an idea of the phenotype up top. So if you don't think that 5% difference in kernel size seems that too different intuitively, you could see a pretty distinct phenotypic difference between McKay and Sparrow. So the last point here, another example, a recent QTL was detected, a single loci, linkage group 1 in the UC Davis program for this trait, and it actually explained quite a bit of variance, so this is something that we'll target. Looking forward, last slide here, anthracnose resistance. So Sparrow by Schessler provides some good opportunity at our site. Sparrow shows to be highly tolerant. Schessler less tolerant. In some years, we'd even go as far to say weakly tolerant. See the phenotypes up top. We did some initial phenotyping based on a class-based index this summer, and the phenotyping was done by summer. And you see the segregation pattern here is pretty ideal for QTL mapping, a nice normal distribution. We'll go forward with a few other phenotypic methods. We'll use this visual class rating to do our association analysis. Then we'll also do some more quantitative measures where we do leaf inoculations with a needle on each leaflet, looking at necrotic size so that we can maybe get a more high resolution distribution for QTL mapping. And then what would be a whole different presentation would be the work with Chung Ho, bringing in a whole metabolomics arm to this. Should be multi-stage to identify metabolites associated with anthracnose resistance, and then in vitro identify some that have bioactivity against abating fungal growth, and then using those after a few years of work to then quantify their expression within our mapping population to do metabolite-based QTL mapping, which should give us some opportunity to actually identify causal variants as opposed to wide QTL regions. And that's it.
Okay. So I'm trained as a microbial ecologist, and for this presentation I will be focusing on soil microbial properties of some of the projects that has been going on at the Center for Agroforestry. So first I will expand on what Priyanka presented for her flash talk. Here are a few slides which show the method that she used. So this is the artificial media that she created, marginal soil with biochar. And these are her plants which she showed. And for the microbial analysis component, we used the University of Missouri Metagenomics Center. And the samples were stored at -80 before they were analyzed. And this is the brief method. First, the DNA was extracted, and using PCR, the V4 region was amplified, and the sequences were read using a Illumina MySeq platform. And then they were annotated, and all of this work is done at the MU Metagenomics Center. Based on the results, then we used past software to analyze them, and what we can see here is the numbers represent the different types of microbial OTUs, operational taxonomic unit. So 1 is the soil sample under poplar, 2 is the soil samples under switchgrass, and 3 is the baseline samples. So what you can see here is the total community of the microbial population separated out based on the type of plant species in our samples. So there is impact of the plant species that we can clearly see here. If we look at the relative abundance of the microbial groups, we see some variation. The 4 bars in the extreme right are the baseline samples. Again, I mean, it's not very clearly visible here, I mean, what the differences are, but you can see the colors change a little bit. And we can further look at this difference at a finer resolution, but this is just a representation of the variation that we see. So as far as the conclusion, so biochar addition did not have significant effect on microbial abundance and diversity. Nitrogen application decreased soil microbial abundance and diversity. Again, we don't exactly know why that was the case. Further investigation probably would be necessary. And switchgrass soils had a higher microbial diversity and abundance compared to poplar. The primary reason is switchgrass had a lot of below ground biomass. So these were potted plants, and there was a lot of root biomass in the switchgrass pots, and there were probably root exudates and all sorts of other material available to the microbial population, which probably helped the community in those soils. We are also continuing a second phase of this project where we are looking at impact of saturation at different levels, how that impacts all these properties. We don't have all the data yet, but this was done in a greenhouse, and we had 4 different moisture levels at field capacity, at 50% water holding, a fluctuating water table, and a 100% saturation. So this is just showing the biomass yield. That's all we have at this point. But you can see for switchgrass, there is a declining trend from field capacity to 100% saturation. For poplar, there is a slight increase in 50% water field porosity regime. So probably the poplar was able to utilize a little more moisture for yield at that level. We are also doing a metabolomics component with Chang Ho on these media, and Priyanka is currently working with him, and we will look at the root metabolites of these 2 plant species, and in another few months we will have those data as well. There was another project that I worked with Ranjit. It's an NRCS project looking at microbial community diversity in a farmer field, looking at bacterial and fungal diversity. And the objective was to look at the effect of cover crops and compare it with no cover crops. So this is the bacterial OTU heat map. The 4 columns, if you notice, there are 8 columns. The 4 columns on the left is with no cover crops, and the 4 columns with right is with cover crops. And you can see the 4 columns in the right, the bacterial OTU heat map has a lot more bars. And if we look at the diversity index, we see that in the cover crop, which is to the left, it has a higher diversity index compared to no cover crop soils. So there we can see the benefits of having cover crop in your system. So it increases diversity, and higher diversity means a better soil health and better functioning of the ecosystem as a whole. We also are working on another project where we are looking at effect of land use practices on greenhouse gas emission, microbial diversity. So that project Jamshid presented in the morning. So we are at the very initial stages of that project, and so we will eventually have all of these parameters evaluated for these 3 different systems as well. That's all. Thank you.
Good afternoon. As all of you might already know, that last year our center has conducted a lot of projects on different specialty crops such as pawpaws, elderberry, black walnuts, and chestnuts. And those projects covers different aspects of our different research aspects, such as the production of specialty crops, the analysis of chemical compounds of those specialty crops, and the economics and marketing of those specialty crops. And my research focuses on the economics and marketing of those specialty crops. So the first specialty crop I want to talk about is chestnuts. So this project has lasted for several years, and every year we surveyed chestnut growers in order to better understand the chestnut market. And also we want to provide chestnut growers with the up-to-date marketing information. So in 2019, we surveyed chestnut growers from the American Chestnut Growers Association, and we found out that the chestnut yields in 2018 has increased by 30% compared to the production in 2017. However, the chestnut price did not change. So this is very surprising because based on the economic theory, the increase in supply will lead to the decrease in price. And this result may suggest that although the supply increases, the consumers' demand for chestnut also increases. So now I want to share a business secret with you, with this team. So can I, Mr. Bill Stover? Can I share your secret with this group? So Mr. Bill Stover has sold out all his 6,000 pounds of chestnuts within 9 days online. So this is very exciting. It shows that consumers like chestnuts. And the majority of the chestnuts are harvested from orchards that were at least 15 years old. So the other project is on pawpaws. So this project wants to examine consumer preferences to pawpaws and value-added pawpaw products. So in September last year, our pawpaw team, with the help of many volunteers, has went to 4 different farmers markets to collect information. And our results showed that most of our respondents had heard about pawpaws before. And I'm a little surprised by this number because I think there might not be so many people knows about pawpaws before. But it might because we surveyed our respondents at the farmers market, and those people at the farmers market, they may know more about the native fruits. So that's my guess. And 1/3 of our respondents indicated they had eaten pawpaws before. But most of our respondents indicated that pawpaws has a flavor of mango, banana, papaya, or pineapple. And 80% of our respondents liked eating fresh pawpaws. And for your reference, 87% of our respondents indicated they like eating mangoes. So mango, everybody knows mango has a huge market. So this may suggest that pawpaws may also have a huge market in the future. And right now, what we have to do is to increase consumers' familiarity with pawpaws. And 87% of our respondents would purchase fresh pawpaws in the future. And majority of our respondents, they liked eating pawpaw muffins, which is good. So consumer preferences to organic and pesticide-free pawpaws. Our survey result indicated that consumers like organic pawpaws better than conventional pawpaws, and they are willing to pay 1.7 cents more for the organic products, and they also prefer pesticide-free pawpaws better than conventional pawpaw. And our consumers are willing to pay more than $2 per pound for locally produced pawpaws compared to pawpaws that has an unknown origin. So now I'm going to transfer from specialty crops to cover crop economics. So this project wants to estimate the economic impacts of cover crops on the cash crop production. So we selected a research field in Sheridan County, Missouri, and we divided the field in 6 different fields, with 1 field as the control field and 5 other fields as the treatment fields. The treatment fields added cover crops in a soybean, wheat, and corn rotation, and the control field did not use cover crops. So here is what we found. The control field has an average annual yield which is less than treatment fields. This is good. This suggests that the use of cover crops can increase the yields of cash crops. And we also compared the economic profits. Then the control field has a higher economic profits than the treatment fields. But the good thing is it is only in very short term, and our research only has 4 years data, so this is our results. I guess this is because using the cover crops may bring additional costs to cash crop production. But we hypothesize that in the long term, the use of cover crops can increase the economic profits of cash crops. And we will continue collecting economic data in that field, and then we will test our hypothesis. Thank you very much.
All right, so I am Ashley Conway. I am new faculty here, as was mentioned, at the Center for Agroforestry. And my missive, unlike Ron, who has the wonderful job of building on a research program, I get to build one relatively from scratch, which is exciting and really puts my creative skills to the test. And I really am going to give you today a very high, like 100,000 foot level of what I've seen in the past 5 months since I got here to Missouri. So ultimately, what I've found here is that I want to approach this from the understanding that it's not that silvopasture research has not been done here, including Rob Kallenbach's done work here, Jean Garrett has done phenomenal research in the past. So it would be a mistake to say that we're building it completely from ground zero. We have a strong enough basis in the literature of temperate agroforestry and silvopasture to have an understanding that there is potential for silvopasture. What I'm really running into as I'm talking to producers especially and other colleagues is this is really the big question. We know that there's potential, we understand some of the dynamics involved, but if we want to scale up and make it an applicable practice, how do we do it? How do we go about it? What are the actual management restrictions and challenges that we run into? And what does this do for economics? What does this do for livestock? What does this do for our environment? So as I'm parsing through all of this information and kind of getting my feet under me, I started in August, so I'm not even quite at 6 months here yet. I've really kind of parsed apart these problems of silvopasture into 2 primary categories. And as I build this program, I'm kind of envisioning that ancillary projects will kind of fall under either one or the other of these 2 types of silvopasture. So the first one I'm calling native woodland conversion silvopasture. I've talked to Joe Alley here at NRCS. I think typically in the past, putting livestock in a wooded area, it has been very discouraged, particularly by conservationists, by forest groups, for good reason, because when it's unmanaged and not done properly, it can be very destructive. It's not necessarily good for animal production. It can be very degrading for the ecosystem. But Missouri has so much wooded land, unlike the barren plains of Nebraska where I just came from. And I think that there's exceptional potential here where there are landowners who are small producers or even mid-sized producers that have wooded area on their property, and they are probably, statistically speaking, small cow-calf producers. And they fall into either one of 2 categories. Either they have fenced out their livestock completely from the wooded area based on recommendations, and that wooded land is unmanaged and not necessarily in a healthy state. So the invasive species grow up in the underbrush, but their livestock don't have access to it, or they do allow their livestock access to it and they're not really doing it in a responsible way. There is potential for a third category of people who are responsibly doing this type of silvopasture, but we don't have any numbers on that yet. So my first area of focus is going to be not necessarily prioritizing, but one focus that I'm looking at is how can we encourage landowners that have forested land on their property, if they want to maximize their land base, if they want to increase the utility of their land, manage their forest systems in a responsible way, but also get some agriculture and livestock production benefits from that as well. And then look at really the benefits, the costs, any trade-offs, potentials, and look at how we can optimize the system from a really agro-ecosystem perspective. The second type of silvopasture that I will be simultaneously and concurrently looking at is what I'm calling plantation silvopasture. So this is much more of the type of silvopasture that I think we think of in the literature. It's been in the past easier to control, so there's a lot more research available on planting trees into an area and putting livestock in there and understanding that system dynamic. But I've separated them out because they really are two different beasts. We're looking at apples and oranges here for silvopasture. So there are different management questions, there's different dynamics, and different research potential in these two areas. So my overall program goal, what I'm kind of narrowed in on at this point, is that I'm going to be developing a silvopasture research program that really addresses both applied and basic science questions into these two primary types of temperate hardwood silvopasture, and really focusing specifically on Missouri first and foremost, but then looking for opportunities to expand it in the ecosystem region. So looking east, west, south, our partners down in Boonville, ARS, have a lot of collaboration potential to look at these two types of silvopasture. How can we utilize our woodland spaces to increase silvopasture practice among woodland-owning cattle producers that really either are leaving their woodlands unmanaged or are grazing in an unsustainable fashion? How can we improve this practice? The second component, looking at plantation silvopasture, the improvement of timber production, specialty crops, or highly integrated perennial silvopasture systems on small farms, and looking at trying to answer management questions and ecosystem impact perspective from that perspective. Is that 1 minute left, Summer? Oh my goodness, okay. So at this point, what I really have are just broad goals, so I won't get into too many of these things. I have so many questions. There really is untapped potential for all of the research areas that we could go into, so I'm exploring a lot of different options. Right now, I'll focus on this last slide here, what I've kind of narrowed down for short-term projects and long-term projects. So we have some old pine plots at Southwest Research Center at Mount Vernon that Andy's shown us. I'm looking at renovating that for a long-term project where we can utilize that space to look at plantation silvopasture. And simultaneously at WARDAC, our research center at WARDAC, revamping those woodland silvopasture plots to make, again, a long-term study space. And as we build up, once we have that infrastructure in place to look at livestock from a systems perspective and have that ability to do that, then we'll be able to get into more management questions. And as we build to that, focusing on some short-term projects where we maybe conduct some silvopasture surveys, looking at forage production, like what Amanda Jo talked about earlier, looking at some maybe monitoring studies when we find producer collaborators. I've kind of moving forward on an elderberry fodder project just because opportunity has arisen. So maybe by this time next year I'll have data to present to you. And really this is just scratching the surface. So lots of ideas at this point. I'm narrowing things down, but this is really the primary direction that I'm moving towards at this point. And I look forward to having answers for you at some time in the near future. So thank you.
Okay, my name is Philip Owens. I'm the research leader at the Dale Bumpers Small Farms Research Center in Boonville, Arkansas. I would like to discuss what we do and how we are related to the Agroforestry Center at University of Missouri. I have two scientists that work with me at Boonville. Joan Burke, she works in small ruminant research, mostly sheep And then Jose Franco, we just hired last June, who's an ecologist, and he's working more on systems-based research. We also work very closely with Fayetteville USDA ARS unit. Amanda Ashworth and Philip Moore, they're very productive. Philip Moore won the ASA Environmental Quality Research Award. Amanda Ashworth last year won the Early Career for USDA ARS. It's an award that takes they look at your production in the first 7 years, and it took her about 2.5 years to outbeat everyone else. So she'll be nominated for the presidential award as well. So they're a great group, and so this is really the Arkansas ARS unit that focuses some on agroforestry research. The Boonville Center, our mission is to develop scientific principles and technologies to enhance profitability and sustainability of small-scale farms, which we feel like agroforestry helps with the diversity of small farm systems to help minimize risk and help with diversity of products. Our center has also 110 acres of certified organic land, which I think is very important infrastructure for us, because I feel like organic production is another potentially value-added product that people can add to their system. So we have organic animals as well as organic specialty crop area set aside for research. We have cattle and sheep, about 400 head of cattle, 300 head of sheep. 3 scientists, as I mentioned. We have 3 postdocs, 4 grad students, and 3 visiting scientists from France that will show up in a few weeks. I really push publications and productivity, because that's the metrics that are used to evaluate our productivity within ARS. So we're up at about 8.3 pubs per scientist, and continuing to push that. And when we work with others, we really stress that publications are very important for us. We get about $3.9 million a year annually, and we send about $930,000 to the Missouri Agroforestry Center every year. So it's been nice with Sarah joining and Ron and Ashley. We're really starting to coalesce as a group, I think, and starting to put our heads together to focus more on collaborative research that can be helpful for both of us. As far as our infrastructure and research, we have some loblolly pine. We have about 80 acres that are on 16, 32, and 48 foot spacings. So that's the alleyway spacings. And so Christine Neiman, who's a postdoc, and Jose Franco are beginning some native grass studies, native forages, warm season and cool season forage studies with grazing in the alleyways to look at some different management strategies to try to look at spacing and sunlight and how those forages will produce within the alleys. And I just want to give you an overview of some of the agroforestry research on a couple of papers that we've had published this year and some of the projects. One of the big projects we have is at Fayetteville, Arkansas, working with our group there. Andy Thomas is critical to this group. He's been publishing, sharing data, and shows up and helps work, and so we appreciate you, Andy. But we've got multiple layers on this, and Amanda got a postdoc that's funded to look at big data, because the idea is we're trying to build the data structure, something that's scalable. And so if it's small enough that we can understand the processes and interactions between the multiple layers of data, integrate that, it can be scalable. So we have atmospheric data, so we have eddy covariance towers looking at CO2 flux. We have animal data layers, so we've got GPS collars on the cows so we know where they're at and what they're doing. And linking that, if you can look in the bottom right-hand corner, you can see the soil map. I'm a soil mapper, so I make these high-intensity soil maps. We looked at soil depth. The figure next to it with the green dots, that's where the cattle are grazing. And so every time their head goes down, it gets a GPS point. So we're seeing some really neat soil, plant, animal interactions, and that is being built within this big data tool to try to analyze some of these interactions that we wouldn't necessarily have known without the data. So our plant layers, we got forages, forage quality, botanical compositions, and tree yields. One project I'm working with Andy on is We're noticing differences in tree rates of growth and sizes based on the soils and the landscape, because the more water, the more biomass you can add. So Andy provided some data. We're working with Center for Advanced Spatial Technology to get some high-resolution LiDAR so we can assess the biomass in the roads that go across different areas and relate that to the soils and the water availability. And so we're using Andy's data as kind of the check on the LiDAR data. And we're also incorporating ground penetrating radar, continuous EC measurements, and just lots of data on a small area, but it's helping us understand the system a little bit better. This is quickly, we have 15 small mini watersheds. We've been monitoring for 18 years. We've captured every single runoff event, and we know the load and the, So we know the loads of nutrients, and we have riparian buffers, grass buffers, continuous grazing, rotational grazing. So we're looking at these management influences and impacts on water runoff. And the riparian buffer, we're seeing we're actually getting some quantified benefits of riparian buffers and grass buffers. I see I've got 1 minute, so I'm trying to rush through this. The bristly locust, Amanda and Joan with a postdoc published some results on grazing bristly locusts, and basically found the nutrient content for bristly locusts is equal to some of the forage production. So it could be something that a producer could add as a shade, a nutrient source, as well as integrate within their silvopasture system. And then we're also looking at, there's a national scenic river in Arkansas called the Mulberry River. It's a recreational river. Very little, well, I would say hardly any urban centers in the whole watershed. So it's very clean, but the pH has been dropped a unit over the past 10 years. We're trying to understand why. One of the hypotheses was it's been converting from deciduous to coniferous. We thought maybe it was the leaf litter, the pines acidifying the soil, causing that change that we're seeing in the water. We had a graduate student just publish this paper and really didn't find much differences in the soil chemical properties. So we're looking at other sources like potentially sulfur from coal burning power plants in Oklahoma. Our next hypothesis. I'm wrapping up a big project where I've mapped 4 nations in Central America: El Salvador, Honduras, Nicaragua, Guatemala. We're mapping these at 30-meter pixel resolutions so we can understand the soil properties to build interpretations. Properties are cool, but people want to know, what do you do with it? And so we're condensing that so that people can make decisions on maps like this. So as part of our interpretations are including agroforestry, and Amanda's leading the team on the interpretations. I'm leading the team on digital soil properties. And but putting those together, and we built an app working with Virginia Tech so that you're standing in the field, you can see your soils, you can see your properties, you can see what options do you have on different parts of the field. So we're even down to looking at small hectares where small farmers can utilize this information. With that, I'll conclude my overview of the Dale Bumper Small Farms Research Center. But I'm really excited about the future. It seems like things are coming together better. Again, really happy to have Ron and Ashley on board. They've been reaching out to our group, and I think that's been very positive.
A few people have come in, so I'll reintroduce myself briefly. Andy Thomas. I work in plant science, but I'm based at the Southwest Research Center in Mount Vernon, about 3.5 hours from here. Been there going on 24 years. And this is just kind of an overview, the year of the pawpaw, 2019. So I already talked about elderberry this morning, and Sam did, so I'm going to skip that. I've been growing pawpaws for almost 20 years, and it's just amazing to see the interest increase and increase. And 2019 was kind of crazy. So 2018, this beer was made by Boulevard Brewery in Kansas City, which was really, really amazing. I provided about 500 or 600 pounds of pawpaws to them, and they debuted this beer, which is very interesting. $25 a bottle. It flew off the shelves. They were sold out within a month, and it's a very limited edition beer. So 2019, Bill Rupert knows all about the year of the pawpaw was named the Missouri State Fruit. He can tell the story, but the governor signed it in, what, July or something, and suddenly, of course, my email and phone's ringing off the hook. I got a couple emails from the dean's assistant, James, that the dean and the chancellor want pawpaw ice cream and they want it now. Are they in here? So, okay, well, you have to grow the fruit, you have to harvest it. It's very delicate fruit. Someone has to pulp it. You don't just, and someone has to figure out how to make the ice cream. And so it kind of didn't hear anything for a while, and then it was getting closer to harvest. And a lot of these emails, they may have been coming from Mike. They were coming to me secondhand. Oh, Andy, when are you bringing up all this fruit? Okay. So finally, where's Caroline? There she is. So Caroline really stepped in, and I was harvesting pawpaws. And okay, I can drive this load up here, a few hundred pounds of fruit, and then is it just going to sit in the cold room and rot? It has fruit flies on it. So she arranged, I don't know how she did it, but I showed up. She came, got in my car, and said, we're driving over to Food Science right now. And there were 5 young men standing there, like, what in the heck are these things? And I ate a couple pawpaws and showed them, these are pretty good. And are you serious? And so I left, and then a few hours later, Carolyn was covered in pawpaw juice and pulp. And I saw her, and they had pulped all of this. So the folks there in Food Science arranged these students. The fruit was pulped. It's a very delicate fruit. And that's about the last I heard of it. So I thought, okay, hopefully the ice cream was made. And a month or two later, we had a big event at the Southwest Center, and the dean showed up with this pawpaw ice cream, which is made by the University of Missouri, the Bucks ice cream place. Did anyone else get to try this? It's delicious. It's really, really good ice cream, and it has candied black walnuts in it. I could not believe how good it was. So I kept trying to explain to the dean and his assistant that, yes, I can provide pawpaws, but let's have a farmer provide these. This is a market. And so I don't know where that's going, but now, unfortunately, I have heard that they're wanting to make a pawpaw essence. So either in the lab or some way where they're not using the fruit, which of course the fruit is very variable, and so I'm not sure where this is going, but very exciting development. It's really good. I don't know if it's available anymore, if it was a one-time thing. So not to go on too long, but Jen mentioned the Specialty Crops Grant, which is helping support a lot of this. So any kind of a fun after working on it for all these years. So just briefly on black walnut, things are continuing. Now that Ron is here, I'm really excited. He mentioned the 12 advanced selections from Dr. Coggeshall's work that we have deployed at the Southwest Center. And there's a pretty interesting development, very, very recent with Hammonds, the Black Walnut Group in Missouri, and a gentleman named Jack Cotton who lives in Australia. He's American, and for several years now he's this dream come true guy that wants to plant hundreds of acres and thousands of trees of grafted black walnut. So it's a long, long story, but what's happened, I have a conference call this afternoon to Australia on the way home with Jack and Hammonds about forming a joint venture. So they can plant his trees that he's already paid thousands of dollars for, that Forest Keeling is producing, on Hammond's land where they can manage it. So it's very exciting. It's a little nerve-wracking at the same time, getting these two groups together, making hopefully everyone's happy. So those are going in the ground in 3-4 weeks. So there's a lot of urgency here to. The trees are ready to go, and then there'll be a lot more trees going in the fall. So in 2020, there'll be about nearly 500 trees going in. Now some of those are sparrow and McKay for control. And I just wanted to show this picture. I don't have a very good picture of it, but I bought a tree shaker many years ago. They're extremely heavy. I don't know if you have the exact same tree shaker. It's very heavy, very bulky, and I paid $8,000. I'm sure they're $10,000 or $12,000 now. It works great on pecan, but it badly damages the bark on black walnut. And if you damage the bark on a bearing tree, it's devastating. They can survive, but it just shakes the tree and it damages the bark. So this is, I don't have a good picture. This is a whole different kind of tree shaker. It's a fraction of the price. It's about $1,700, and it has a way of shooting a webbing up into the tree. So you're shaking it from the top, just like that, and it actually shakes better. It shakes the nuts down better. So I'm getting this, and I'm real excited to try this because I quit shaking the walnut trees, which means greatly increased harvest labor in the fall. Just waiting on the nuts to fall is not an efficient way of doing this. So a little bit of something new there. And Philip gave a great introduction to the Fayetteville site. I have just a few pictures. Philip said it perfectly, that we have tons of data. So we plant trees, we cut them down. And so this oak harvest was, I think, 2 years ago. But I wanted to show this picture in the lower right. I'm not sure if those are the sycamore or the cottonwood, but we have a part of it planted for kind of a carbon and biofuel study. And this site's fairly mediocre. I mean, the soil is perfectly normal, nothing really good. So that was the first year, 2014. 2 years later, now that student is a little short, but not too short. These are the cottonwoods, 2 years. And remember, when we planted these, we pounded cuttings in the ground with a hammer. So they weren't even rooted yet. So this is 2 years. And then this was last summer. The trees grew so fast, we had to do a thinning, and they've grown so fast, we're going to do another thinning probably this summer. So all this thinning creates data and a huge amount of work. So I've just got a few pictures. So this was last summer in July. So this is how we traditionally did it. We had this tripod thing that when we harvested the oaks, it was pretty easy to chop them up and weigh them. We're getting all this weight data. But you can see here, this is that blue thing. This is a load cell that can weigh. But you can see the tree is dragging on the ground. We weren't able to get very good data. We did the best we could, but we had to bring in the big gun. So we had to bring in this giant tractor, and we were able to see the load cell on there, get our data. So that worked pretty well. Just a picture of all this data being taken, tree height. And then this is one thing we did, which is pretty interesting. The tree compartments were divided into different, trunk, large diameter, small diameter. So anything less than, I don't remember, like 1.5 inches got shredded. We rented a shredder, and this is how we collected the shreddings, and all of that was analyzed as well. So that I did a lot of that, and I sneezed and coughed for several months. So I will not do that again without a mask on. And Philip also showed a picture of this super incredible micrometeorological flux station down at the site. So it's just really a neat multidisciplinary site. So let that go over.
Okay. And this is really just highlights. There's so much more, so if you're curious about any of this, find me afterwards and we can talk. And we heard a little bit about the Agroforestry Academy from Sophie Mendelson this morning, if you were here. She's the MS student who has been doing a really comprehensive evaluation of the Agroforestry Academy. For those who are not familiar, this is a week-long intensive for both as a Train the Trainer program and also for farmer landowners. And this year, we had 30 participants. That's including 4 of the mentor instructors who were with us throughout the week. About half of those were natural resources professionals, half were farmer landowners, to give you a sense of the where everybody fits. So this week-long program, we try to make it as engaging and as exciting as possible because it's a lot packed into a week. So we have seminars, field visits, out to Bill Stouffer is one of the farmers who welcomes us every summer, so thanks, Bill. And we work with a case study farmer every year so that everyone who participates in this program has a chance to work through the planning and design process to help them wherever they're going. And I think this we collect a lot of evaluation data from this program, but one of the things that I think is particularly interesting is this question: how much did each of these topics meet your needs? So I don't know if this is too small for some in the back, but farm visits consistently rank #1 as the most useful thing that people get out of this. This year, forest farming, the training manual, silvopasture, and the planning handbook were in the top also. And so that's the segue into the field events and workshops, because I think the reason why those farm visits tend to yield the most in education for these participants is because it's a time when they can really see and do and sometimes even have hands-on experiences. So we try to do as much of that as we can with public outreach programs. So this year there are sort of 3 main directions that we've been going based on some of our partners. NRCS, we've been working with Joe, Allie, and Lauren Cartwright, and doing a series of specialty crop workshops for farmers and also training programs for the NRCS staff who can then help. This is the idea, right? To grow the network, and the Agroforestry Academy is a big part of that too, so that there are people in the field, not just here at the Center for Agroforestry, folks whose job it is to go out onto these sites to help landowners with the technical assistance piece. We are continuing the USDA SARE Professional Development Program for high school ag teachers. We have 1 more year of that, and reaching out more directly to the teachers and students for them to get the extra support they need. The last 3 years we've been hosting 1 or 2 day workshops for teachers to learn how to use an agroforestry curriculum in their FFA classes, and just noticing that 1 or 2 days probably isn't enough for them to feel really confident with that material. And Jen mentioned the specialty crop, and Andy both mentioned the specialty crop block grant for pawpaw. So we've got some energy building around pawpaw, and to, I think, really complement Jen's economic and marketing work to raise public awareness about how delicious pawpaw can be. And just a number of other research center field days, including the chestnut roast, and mushroom cultivation is always really popular, so we try to meet demand as we can. Thankfully, Sarah Havens, she's a natural resources extension person here who's mostly in the southern half of the state, and she's really taking on a lot of mushroom cultivation workshops this year. So gratitude to her for that. A couple new things. Well, one new thing and one not too old thing. The podcast Sarah mentioned. This is something that I took on as a little side project, and it's turned into a bigger project. But it's, for as little as I've been promoting it, I've been really amazed just how many listeners are downloading these episodes. So I try to get one out each month. And has anyone had a chance to listen to the podcast? Just a few? Okay, well, check it out. It's on all podcast apps. So it's syndicated through Libsyn, Liberated Syndication. So you can find it on iTunes and Stitcher, or whatever you use. I honestly don't know as much as I probably could about the tech side of things. But I'm putting the content together, and a lot of that content we collect throughout the year at the education programs that we host. So anytime, like today, we're collecting audio during these talks so that we can use some of the really great nuggets of what you all have shared in these themed programs. And the idea with these, the way they differ from the webinar series, is that they're pretty short. They're half an hour long, approximately, and they're really accessible. So someone who's never heard of agroforestry can still listen to one of these podcasts and get sort of a sense of what's happening. Whereas the webinar series, we're getting a lot more in-depth. So these are generally hour-long seminars from partners and people internally to share more about their work. So I think they really complement each other well. One of the things that I'm really excited about going on here now is that Sarah also shared at the Land of the Osages Research Center, the newest, whoa, 1 minute already. Okay. The first new research center in over 30 years for MU, and an incredible opportunity for us to partner with the Osage Nation. So at this time, I just want to offer a land acknowledgement. We are on the ancestral homeland of the Osage. Their ancestral name is Wajaje or Newtachi, and also the Oto Missouria. They've combined since colonization, but they were distinct peoples. Prior to colonization, their names were Chiweri and Unoka, or Niukanska is the other Osage name. So it's an honor to have the chance to work with them. As Sarah mentioned, agroforestry is really rooted in indigenous practices, so this makes sense for us. It makes sense for a lot of people to start doing land acknowledgement, but especially for us here at the center. And as a personal passion and also this tremendous opportunity at LORC, it's important for us to think about developing a research protocol that's culturally relevant. So just like we have the IRB here for doing research, we have to make sure we're following the university's protocols. When you do research with a tribal community, you have to make sure to follow their protocol too. And they often have their own unique protocols depending what community you're working with. And because of a history of extraction, of information and resources from these peoples, it's especially important that we consider how we can make sure this research really serves that community too. It can be mutually beneficial. So I'm working on a PhD now, and this is the focus area. So just to give you a sampling of some of the many other things we offer, of course, the Agroforestry Symposium was yesterday. Hopefully you all were there. And we also piloted an undergraduate student summer internship this past summer, and she's in the top right corner there, Aiden Cornelison. And that went pretty well. It would be great once we have more facilities at HARC to be able to host more interns there and have a more robust program. And I also have a passion in youth. If anyone was at the symposium yesterday, we heard from some people talking about the importance of youth education. This understanding of agroforestry has to start young. So I work with the Missouri River Relief Organization, the Natural Resources Career Academy, and a few other groups to make sure that we're hitting those younger crowds too. They're important. We've got some new HARC demonstration areas. Of course, the e-newsletters, Green Horizons and Action in Agroforestry. If you're not already signed up for those, tell Mike Gold, and he'll put your email on the list. And really excited about the new website design, which will make sign up for those newsletters much easier also. Okay, looking forward to 2020 as my last little piece here. I would really like to be thinking about better evaluation tools to measure impact. All of this that we're doing, what kind of an impact are we having on people after they leave these workshops? Partnerships are so critical for outreach. You know, there's only one of me, but thanks to our partners, NRCS is a major one, we can have a much greater reach. And the continuity of this education program. So the youth aspect is part of that, and really glad that we'll have some undergraduate courses to offer this coming year. So there's lots of exciting stuff on the way.
This project was to evaluate various conservation management practices on water quality. It is a NRCS funded project conducted on private land on a property. That's why you don't have direct numbers. For example, if you are talking about sediment, you say how many kilograms, how many tons per hectare. We wanted to maintain the privacy, so we have converted those values to mean ranking. As you see on the figure, top figure, you can see wheat and corn when it was left on the ground, that had the lowest sediment loss and the nitrate loss. So when you are using avoid trap control mechanism, lower figure, that's when you get the most of the benefits in terms of reducing nitrogen, sediment, and phosphorus. So avoid is kind of a nutrient management plan. Farmers can apply different types of nutrient management. Trap and control is buffers, waterways, all those kinds of measurement, cover crops. So some of the farmers, they apply all these methods. Some farmers said grass waterways, cover crops, crop rotation, buffers, and everything. So those farmers reported the lowest loss. Actually, this study was started in 2011. So we completed a paper and submitted to Journal of Soil Conservation last year in October, November. Steve Hefner from NRCS is a co-author, Karen Brinkman. Shibu and I started the working on the project from Missouri, from MU. Then this project, we were looking at soil microbial properties using PLFA at Greenlee. Those dots represent sampling location. Yellow is crop watershed, only corn, soybean. Then the middle watershed is agroforestry. We took samples from the tree areas, and green represents now biomass. We converted those two watersheds to biomass. From 91 to 2012, it was under corn soybean rotation. Now green means biomass crops, so we were able to compare microbial properties using PLFA for biomass, grass buffers, that's blue lines on the west watershed, on the middle watershed trees, and the crops. What you see here is, if you look at the third set of columns, row crop had the lowest total microbial biomass and the lowest soil organic carbon, which clearly says when you have perennial vegetation management systems, grass waterways, grass buffers, biomass crops, different distances from trees, they had higher total microbial biomass and soil organic carbon. Then we can look at all the total bacterial biomass, gram plus, gram negative, fungi, actinomycetes, saprophytes, different fungi, protozoa, and rhizobium. All these figures show third set of columns, that's for the row crop, had the lowest concentration. So when you have buffers, it increases microbial diversity in the soil. On the previous talk, I showed you antibiotic degradation and so many soil reactions when you have diverse microbial communities. So that means nutrient cycling, carbon sequestration, degradation of chemicals. This will be published in February issue of Agronomy Journal. This is a cover crop project that we did, this at Chariton County. It was a NRCS funded grant. We had soil moisture sensors at 3 different depths, 10, 20, 30 centimeter depth, compared soil moisture changes from January to August for 3 consecutive years, from cover crops areas and no cover crop areas. So what we saw was cover crop areas tended to store more soil water in the soil compared to no cover crop. So there is a big issue because farmers don't know when to terminate the cover crop. If you do not terminate at the right time, especially during drought years, the cash crop, corn or soybean, won't get enough water. So farmers need to know when to terminate the cash crop. So this shows if it is a drought year, you have to terminate cover crops earlier. If it is a wet year, you can postpone terminating cover crops. Usually farmers kill cover crops about 10 days, 12 days before they are going to plant corn and soybean. So they can decide the benefits of cover crops and when to terminate. This is kind of a summary, 0 to 10 centimeter depth, 10 to 20 and 20 to 30. Gray lines represent for cover crops. For all 3 depths, cover crops had higher moisture content in the soil. Maybe due to several reasons. Carbon, increase on soil properties, and rooting patterns. And for this study, we took soil samples from the same Chariton, we did it at Bradford, cover crop and no cover crop. We took small soil cores about 2 centimeter diameter, 4 centimeter long. Took to the geology department. They have acquired a X-ray CT machine. You can measure soil samples at 29 micrometer resolution. That is about 1/30th of a millimeter. So when you take that many images, within a 4-inch core, you can get about 4,000 images, cross-sectional images. So that means we can compare pore continuity, pore distribution, all the patterns compared to the normal X-ray machine at the hospital. So what this clearly shows, cover crop soils had higher total porosity, and we categorize porosity by micrometer size, less than 50, 50 to 100, and greater than 100. So all soils had higher porosity with cover crops. And this is the summary which shows the distribution of porosity by depth. So connected line is for the cover crop, As you see, with no cover crops, porosity is very low. With cover crops, it can significantly increase soil porosity. So this shows when you integrate cover crops into cropping practices, it helps improve soil properties, soil bulk density, porosity, water retention, water supply capacity, carbon, and so many other microbial properties. That's all. Thank you.
So the first presentation, I'm going to highlight some of the bioremediation project that has been done at the center. So agroforestry, we do believe agroforestry provide perfect opportunity, to turn the system into the green lever to detoxify that pollutants in the environment. If we can put those components in the right place, right time, we can rely on the mother nature to take care of some of the human the problem. So this wonderful project was originally funded through the NIH, and there's also the NGO they call the Environmental Use Group in Pennsylvania. So we realized that in the hydraulic fracking operation that require more than 750 different chemicals pumped into the ground to maintain those cracks. Okay, and in the rural U.S., more than 15 million Americans living within 1 mile of a fracking operation. And so far, there are more than 82,000 fracking wells has been built since 2005. Imagine that's health implication for the community living nearby that operation. Not to mention what the environmental impact and risk to those wildlife. So my lab, this morning, Mike mentioned that we love working with brewery. So over the years, my lab has developed several methods to quantify those pollutants and their degradation products in water, sediment, urine, fecal, serum, and feather. We took advantage of our infrastructure and worked with the industry, private sector, NGO, and health organization to assess the exposure of the general public to those chemicals and relate that with some of the health risks. And we're expecting to receive another one major grant from the NIH to continue this line of research. Also, at the same time, our group has been working on the idea of using the construct wetland to remove the PDCP, which stands for personal care product and pharmaceutical, in the municipal wastewater. City of Columbia is concentrated with at least 9 hospitals. Where do those personal care products, pharmaceutical compounds go? They go to the drain, right? Or toilet. They end up in the municipal wastewater treatment plant. Because the body only take up 5%. The rest of 95% go down the drain. Also for regular community, usually nowadays people tend to take a lot of like birth control pill, sometimes antidepressant, okay, or anti-inflammatory drugs, antifungal, recreational drug, anti-seizure, the steroid or compound that lower the cholesterol. They all end up in the wastewater. So who is the first victim? Fish. We realize that almost more than 95% of fish that has intersex symptoms, which means as both female and male reproductive, they found nearby the discharge of municipal wastewater. treatment plant. So over the years, our lab developed a method we can quantify. Right now, we're monitoring 85 compounds in the wetland system. That based on what we have learned, more than in general, more than 80% of these personal care products, pharmaceutical, they can be removed by this constructed wetland system. So in our reports, we are taking advantage of what Mother Nature can offer, get rid of these pollutants in the environment. Because a lot of pollutants, they behave like endocrine disruptor. They only require very small level of this compound that trigger the biological function. Right now, I'm moving to the direction of water informatics. So I have a vision. Since we already developed very sensitive, robust screening analytical method, right now I'm working with several faculty on campus. We're trying to work on proposal idea. What if we could just take droplet water, we can assess what kind of personal care product is in there, then we can evaluate the overall well-being of the community. For example, if we see a high concentration of antidepressant, birth control pill, or recreational drug, that kind of suggests this community might under some kind of stress. It could be financial stress, family or work-related stress. Okay, I think that's more accurate than the conventional survey approach. Because no one can hide it. Okay, and eventually we would like to develop the model just by analyzing one drop of the water. You can predict not only the community well-being, also we can predict what's the economic opportunity in the community and related. Hopefully we can, I propose a feature. Hopefully just by knowing those chemical profile, we can predict the future stock market. That's the kind of the vision we're proposing by integrating the chemists on campus, economists, and also the social scientists. Okay. So this is my first. Okay. Also, this year we're fortunate our center become a part of this Great Lake restoration initiative. This is the one that the major EPA restoration project over the past 20 years. So EPA provided $11 billion to sponsor 5,000 projects trying to minimize or prevent any leachate from getting to the Great Lakes. So our role is our collaborator from Forest Service and this, they are working on a system to utilize the powder tree as the remediation approach to prevent this contaminant reach the Great Lakes. Am I over time?
5 minutes.
Okay. Any question? Now I'm moving to. Okay, so now I'm going to move into this phytochemistry project. Yeah, one thing nice about working in academia, first, we have that freedom to explore. Also, at MU campus, they offer so many disciplines that allow us to put together to accomplish our vision. So for me, I feel very fortunate to be able to be part of a family. Okay, so for phytochemistry, we research, I only have two objectives. One is identify health promoting compound. Another objective is to explore the new application and niche market for the industry. Right at this point, I'm particularly interested in cosmetic and personal care products. And over past 2 years, we have been receiving the funding from the state specialty crop grant. We received three funding. One is to sponsor our research on phytochemical profiling on black walnut. Right now, we are screening and profiling the phytochemical among 24 varieties, cultivars of black walnut. Also, we receive funding to study the chemical profile and the new application for the elderberry. We are working on 21 different varieties by working with Andy. Also, last year, we also received the funding through the same specialty crop program to study the phytochemicals on 12 different varieties of pawpaw. At the same time, with the support through USAID, we had enough manpower to also to study the phytochemical profile for the biofuel feedstock, including the 6 different varieties of switchgrass. Also, our lab has been working on the taking advantage of low-value material and try to explore opportunity to turn that into raw material for the industry for Osage orange and spent coffee grounds. Okay. So in order to identify the niche market and figure out what are the molecule we can take advantage of, so we integrate the 2 major facilities. One is the MU metabolomic platform. Another one is the high throughput screening facility at the University of Kansas. Yeah, I will not get into too much detail, but basically, in order to identify and profiling the chemical, we have to heavily rely on the fingerprint of each molecule. So every 15 minutes, we will generate about 250,000 of a fingerprint. Then we compare this fingerprint with a fingerprint in the library to identify those compounds. Then once we isolate a compound, we send those purified chemicals to the high throughput screening. With those high throughput facility, they can process 300,000 bioassays in a single week. So using this approach, we can easily identify the bioactive compound and their application. So our students have been working really hard to compile their list, and also we share this list with every collaborator. And by brainstorming, that will help us to find the niche market. At the same time, we have been working with the food processing engineer at the MU campus, also another collaborators at the University of Pretoria, specializing the cosmetic formulation. There's a reason we are interested in the personal care product and personal care product cosmetic. First, if you want, for example, the landowner want to start up the business. If they want to take the products to the market, they don't need to go through all the FDA regulation, so they can immediately generate the revenue. Okay. Secondly, cosmetic product, you can sell that for sometimes up to $20 to $30 per bottle, and basically 95% are water. So only 5% are active ingredient. I think that's a unique niche for the small operator to start is to start building up their business, start seeing the revenue. So, but first you have to find a niche. If you try to reinvent the wheel or follow someone's footstep, it's not going to work. That's why our center is providing scientific information to help them facilitate that process. Okay, this concludes my presentation.